Good evening, dear colleagues. It is my pleasure to welcome you again on our Ovogene Egg Bank webinar series. It is already a great tradition to meet every month, and we already discussed such topics as donor recruitment, legal aspects, inclusion and exclusion criteria, oocyte selection criteria, and our unique technique of genetically certified oocytes. And our today's topic, it is medical and embryological KPIs for oocyte donation and egg banking. My name is Dr. Uliana Dorofeyeva. I'm a medical director at Ovogene Egg Bank. And this evening, I will talk to you together with my great colleague, senior clinical embryologist, laboratory manager, and scientific advisor of Ovogene Egg Bank, Birol Aydin. So let's start talking about medical KPIs for oocyte donation and egg banking. I will share my screen. Just a minute. Opening. Please let me know in the chart that you can see my screen. I will be really grateful for this. And before we start, why our team is using the KPIs? It is, of course, to monitor our results, to measure the progress, to analyze the patterns over the time. But also, our quality management system is developed and certified according to requirements of ISO 9001, and we ensure the compliance of all our processes and materials with international standards for biological material processing, from donor selection to transportation of biological materials. And main quality management system procedures cover all the critical aspects as a donor selection and screening, scoring, cryopreservation, sewing, packaging, and storage of biological materials, as well as identification, labeling, and traceability for materials, equipment, and materials which are used, laboratory equipment quality control of working environment, non-conformance management, regular audits and risk management, management for documented information, staff training, and constant improvements. Due to the fact that we comply with all these regulations, we are proud to be fully compliant with the EU regulations for reproductive cells, HFEA regulations, FDA and Health Canada, tissue regulations, Reproductive Technology Accreditation Committee for Australia, and such scientific organizations as ASHRAE, ISRM, and Red Lara for donor selection, evaluation, and testing, producing, storage, and transportation of biological materials all over the globe. And Ovogene Egg Bank team use KPIs to evaluate our progress toward reaching targeted goals, such as variety and quality of our donor materials, compliance with internal quality control requirements, and external quality assurance, achieving the highest possible outcomes with our partner clinics globally. Due to the fact of the strict selection criteria for our donor candidate, only 3 out of 10 candidates will become our donors, and the process is arranged by the following algorithm: that 1/3 of those who applied to be our donors will not be accepted after personal, psychological, and social evaluation by our professionals, or will refuse after learning all details about the program, because as we all know, egg donation is absolutely voluntary process. A little bit less than 1/3 will not be accepted due to medical reasons, and these medical reasons are mainly initial ultrasound evaluation and the gynecological evaluation as accessing of ovarian reserve and antimural hormone level. Some will not be excluded after infection diseases screening being positively tested. some excluded after genetic or chromosomal screening, genetic consultation, evaluation of the pedigree and medical history of the donor family, and only 1/3 will be accepted as a candidate for further stimulations. It is very important from the medical point of view to bring your attention to the age of the donors, as we are going to talk about medical KPIs, and we need to understand the difference between the cohort of the patients we usually see on our consultations who are infertile with different complications and the solutions we need to find for them, and compare to the donor cohort. And the main age of the donor is between 18 to 29 years old, meaning they are all young and healthy. In Ukraine, the donors are even with the proven fertility, which also proves their results of the stimulation and the outcomes. Of course, they have no hereditary habits, appropriate ovarian reserve. If we are talking about figures, so the inclusion criteria for IMH will be over 3 picograms per ml, and over 16 antral follicles in both ovaries for donor to be included into our database. And it should be dependable, mature, and responsible person. In order to evaluate the profile of our donors, we did a comparison between our database of the donor 10 years ago. In 2010, we evaluated over 600 profiles and more than 2,500 profiles in 2020, which was last year. And we have seen also that an average age of our donors was also a little bit increased. This is the trend which we see with all our patients. And this solution we need to find for all those who are over 40 years old looking for the treatment, for the successful treatment. But again, bringing your attention to the average age of our donors in our database right now is 25 years old. And average IMH was not calculated and monitored 10 years ago, so we cannot compare, but right now this is over 5 picogram per ml. So there is a clear difference between two cohorts of our regular patients who are over 30 years old, sometimes over 35 years old, often with gynecological or combined pathology, with decreased IMH, with potential genetic disorders, and young and healthy, less than 30 years old women, only good responders with no gynecological or related pathology, and even more with the proven fertility. So the target for stimulation protocol is to be the one which will produce a homogeneous cohort of mature and high quality oocytes, optimal number of well-expanded cumulus oocyte complexes, almost from each follicle over 16 millimeter in diameter. And our standard protocol, this is antagonist protocol with agonist triggering, so it is absolutely safe. So the next aim of the protocol is to be with no inconveniences or risks for the donor. So the KPIs themselves are calculated per started cycles, and we evaluate percentage and number of the canceled cycles according to the number of OPUs. OPUs resulted in bleeding and cycles with OHSS, but I can say for sure that we haven't seen any OHSS since last 3 years until we started to use our standard protocol with agonist triggering. Number of cycles with no oocyte retrieved. Usually, sometimes this can happen, very rarely, but due to the fact of the mistakes by taking medications by the donor, our donors are not using contraceptive pills, so there is almost no issues with no oocyte retrieved after the agonist triggering. According to the number of follicles observed, we calculate number of oocyte harvested, and this is average number of oocyte retrieved per cycle. And you can see 3 parameters in different colors. They are desired, acceptable, or minimal or maximum. So we like to be in our green or yellow field. And if we are on a red field, This means that we need to evaluate the stimulation of this donor, and we do on our medical meetings, and we will change different protocol, the dosage, the attitude to that donor, and we would decide, should we include her for the further stimulation protocols, or should we change something, or should we exclude her from the further treatment. Number of oocyte harvested per number of follicles observed, mature oocyte, but not only mature oocyte per cohort, but also used oocyte. As we have presented, we divide our oocytes, retrieved mature oocyte, into 3 categories: quality 1, quality 2, and quality 3. And for the oocyte banking, we accept only quality 1 oocyte. More in details, Birol will talk later about this. And of course, quantity is important, as we evaluated different parameters of our KPIs, and we consider cycles with less than 12 mature oocytes retrieved in the cycle as non-optimal, and we would definitely evaluate such a donor. How should we change her treatment regime? Should we include her for the further processes? As well as the cycles with more than 30 oocyte harvested in one OPU. There is no aim for us to overstimulate our donors, but of course we need to understand, and we talked before about that this is a cohort of good responders who potentially can respond with more than 30 oocyte collected. But in order to evaluate the quality issues related to the number of oocyte retrieved per cohort, and this is still ongoing discussion in all the publications and literature and conferences, so we divided groups of our donors into the group by number of oocyte retrieved. And there were 4 groups from 1 to 11 oocytes, 12, which is non-optimal per our parameters, between 12 to 24, 25, 30, and over 30. So what we have seen, that there was a trend in decreasing of percentage of mature oocyte per cohort and used oocytes per cohort. But after we qualified the oocytes and considered them for further use, there was no difference into the percentage of fertilization, used blastulation rate per day 5 or 6, and total used rate per whole cohort. Another clinical parameter, this is cumulative clinical pregnancy rate per donated cycle, which is considered as the number of pregnancy or live births we have been achieved from the first until the final treatment cycle, until all embryos were used or patient got pregnant, assuming was no patient dropout rate. And we can clearly see that if more oocyte we consider per cohort being used per patient, higher probability of clinical pregnancy rate per initial embryo transfer and cumulative clinical pregnancy rate per transfer for the patient. Also, we evaluate clinical parameters for our partner clinics, and all are sharing their results, clinical performances with us, as fertilization rate, of course, survival rate. This is what we are tracking nonstop. Fertilization rate, cleavage rate, utilization, and the blastulation rate, clinical pregnancy rate. Results are different for different clinics, and they are evaluated both by clinical professionals and embryologists in order to achieve all target in green. But as this is a mutual cooperation between the clinical professionals, IVF doctors, and embryologists, Birol will continue about embryological KPIs and the management of the oocyte banking. And so far, I would like to thank for your attention. I would ask you to write all your questions into the chat, and we will happily answer after we will finish our presentation. Thank you so much. Please, Birol, your turn.
Hi, welcome everyone to our Ovogene Egg Bank webinar. So as Dr. Uliana explained, of course, we are following very strictly KPI for the egg banking till donor come to oocyte pickup. But when donor come to oocyte pickup, as you know, the main product will be created in the embryology laboratory. So we should also strictly keep quality parameters in embryology laboratory. So how we are organizing this? As a first thing, quality control and IVF lab management for egg banking. So we have to organize it very well, and we have to be sure our supplier laboratory will have a good performance, good fertilization rate, good vitrification and survival rate, and also good blastocyst rate, so which we can control all egg donor as efficiency. So most important things as a morphologically donor oocyte selection. So how supplier clinic for egg bank is choosing egg donor and donor eggs for the morphological assessment. Tissue bank witnessing is a really important, of course, as a supplier clinic, we have a many donor function per day. Of course, still there are many different embryologists are working for the different process, for the different donors in the same time. So there are a big crowd day in the laboratory, and witnessing is a really important for us, how we can keep a traceability. So laboratory audit system, still we are sending all around world frozen donor eggs. And for us, very important to keep all laboratories, all clinics which we are sending donor eggs under optimized standards and optimized protocol. So we are trying to keep very good laboratory audit system all the time. So advanced technologies, of course, now egg banking is a big trend, especially after the COVID pandemic. So now we are trying to increase, we know our responsibility, and we are trying to increase the quality. And in IVF, always you can increase the quality with the technologies. Genetic technologies in a donor egg banking, of course, this is a new trend. Still, all egg banks are working with carrier screening, expanded carrier screening, and many different genetic tests to egg donors. But we were thinking how we can increase this level directly with the biological material for the donor oocytes. So how we are doing a quality control system and lab tracking. So we are doing that totally with a daily digital control. So each embryologist has a special application in their phone, which is connected directly to the tracking system, digital tracking system from the laboratory. And we are able 24 hours to control all parameters in the embryology laboratory. So laboratory humidity, carbon dioxide level, pressure and temperature all the time is updated through our application and phone application. Also, we can monitorize during working time with the special alarm system, emails and SMS systems during the working time in laboratory and when we are out of the laboratory. So also tracking system is connected to incubators as measuring carbon dioxide, temperature and pH control. Also really important, like to measure the real particle control in the laboratory while directly affecting the outcome for the because it can increase the toxicity and can drop the IVF outcomes after egg donation cycle. And of course, oxygen level control is very important while we are working with the liquid nitrogen and is very important for safety of embryology team. When we are talking about the lab tracking, there are three different topics which we need to talk. So we should track incubators because incubators keep still culturing oocyte from the egg pickup till vitrification. Work area, it's really important the laboratory environment should be totally optimized while we are working with the egg donation and a different process in IVF. And also fridge, which we are keeping a media for vitrification and other steps for egg pickup and other. So that should be optimized. How we are doing also pH control for each incubator. So we are using for each incubator special pH measuring system. So it is measuring automatically and monitoring to us. During the culture, for the embryo culturing, always a 6 day of the culturing. Of course, during the oocyte cultivation. Also, we can get from each part of the incubator, each well of the incubator, exactly carbon dioxide level and temperature level by the lab tracking system and phone application. That is most important things, how we are controlling a cryo tank management, because there will be a lot of different donor, different profile, different number. Also, of course, supplier clinics still have different IVF process. So how we can control and how we can easily manipulate to receive frozen material from the tank, and how we can control this algorithm. Of course, we are working directly with software system, which is controlling to cryo tank. Also, we are working with cryo storage, which is connected to data logger, and we can get these data all the time, exact level of the nitrogen, exact temperature of the nitrogen, because the system is working up and down, and there are many different donors inside of cryo tank. So also this connected directly to phone application, which even if we are not in the lab, so we can see any kind of changes for the temperature and for the level of the liquid nitrogen. Well, you see on the left side a tank mapping. It is a special software management system for the cryo tank. When you see the green and red, so when we receive some donor eggs to send to customer to ship, so directly system make them red, and we know this donor doesn't have already material, or just only if there is a 10 stroke, we just release 5 stroke and still there is available 5 stroke. So all this one we are controlling directly with the software management. How we are doing the morphological assessment for the donor egg quality for egg banking. So first thing we take care of, cumulus oocyte complex morphology. So as you know, there is not direct effect according to publication and research from the cumulus oocyte complex morphology to fertilization and cleavage rate, but still there are outcome which directly can affect the blastocyst rate because of the toxicity effect. Zona pellucida, of course, I will talk in the next slide, how we are evaluating zona pellucida and why zona pellucida is really important for morphological assessment of the oocyte. Perivitellin space is really important for us, while large perivitellin space and non-perivitellin space shows really low survival rate after the warming. First, polar body appearance, it is also important. Of course, we can freeze only the mature oocyte and with high quality of the polar bodies. Oocyte shape, if we have different shape of oocyte, which is not usual, so we are not considering for egg banking. Meiotic spindle location, we start to use it directly for as a part of morphological assessment for all egg donors. We are first detecting meiotic spindle, and then we decide if meiotic spindle is visible to freeze this oocyte for egg banking. Granulated cytoplasm, of course, if we have any kind of problem on the cytoplasma as a granulation, also I will talk in the next slide, which artificial intelligence can detect by the fiber optic system, and we can just evaluate this and to get out of egg banking. And of course, time-lapse activity. This is also unique for Ovogene Egg Bank. So we will be able to give to all of our customers like a live time-lapse video for the vitrification during the process, and also their egg quality and morphological assessment. So of course, there are different kind of morphological assessment which we can use, especially for the donors. Mostly they are giving more number of the follicle, especially ovogene egg donors have really good number of follicles. And of course, we cannot receive from the all follicles high quality of oocyte. Still, there are strict parameters which we can use for egg banking, which is not inside of these parameters, that they are going directly a fresh donor cycle. Let's talk about the traceability, how we are controlling traceability and witnessing for the donor egg banking. Of course, this is a very important topic, and we are trying to control that by the electronic witnessing system, not only just human witness system. So we are using that all steps, RFID chips on the petri dishes, straws, all biological materials, blood, and all part of egg banking process. So how electronic witnessing system is working for the donor process? Actually, system is starting directly from the donor stimulation. When doctors stimulate the donor, and when this information comes through to our software management system, so automatically witness system take this information, and when egg donor start to come to function, so egg donor get a special rice band, and from that process till vitrification, end of vitrification, all process controlling by electronic witnessing system under RFID chip. Of course, here also we can control as a staff, each embryologist, their all performance during the vitrification, egg collection, denudation. So how many egg they denudate and how many was survived. How was their morphological assessment? How many oocyte they freeze and how many oocyte from their freezing survived. We can control through electronic witnessing system because system has also software part. We can keep all this data as a server, also in our database. Also, we can control that each embryologist by the special phone application. So when donor come to the operation room, firstly, donor has a special rice band, which they have all information about age of donor, code of donor, date of stimulation, and different information. When they read the chip, so all information is going to main screen and also is going to each tablets which we have in each laminar flow for the embryologist. So during the process, embryologist can control all the process from the tablet, and IVF specialist can control all process during egg pickup from the main screen. So when we start to collect egg by the IVF specialist, number of eggs will be identified by the embryologist, and all this information will go directly to main screen. So in this case, during the oocyte pickup, let's say IVF specialist will pick up 50 follicles. IVF specialist will be aware each follicle, if they find oocyte or some follicle was empty, so all process will be under control. When process finish, as you see in the first image, embryologist just will put a special RFID chip on the petri dish, which we will cultivate oocyte from egg pickup till denudation. When the denudation starts, embryologist just will read the RFID chip by the culture dish, and we will get all information of egg donor on the tablets. So when denudation finish, through tablets, embryologist will make the first assessment of oocyte maturation. So how many mature oocyte we have, how many metaphase 1 we have, or how many GV or degenerative oocyte we have. So system will keep this data and will send the main software management. We will be able to keep all information till oocyte freezing. When oocyte freezing start, first embryologist will read the culture dish, oocyte culture dish, and we will identify the egg donor, and then each straw will have the smart RFID chip, and we can also witnessing for each straw or cryo device by the RFID card reader. About laboratory audit system, first of all, we are analyzing by senior embryologist all KPI from the customer clinic lab. So we are sending them special KPI form, which we have different question. I will show the next slide. And according to that, we are giving our advice what they need to do for their lab to get a better outcome for egg donation cycle. So analyzing of quality objectives, let's say fertilization rate, cleavage rate, the blastocyst rate, and embryo oocyte survival rate, we are asking from the customer clinics to find out if there is some gap in some step. So analyzing of quality indicators as type of culture media, cultivation technique, and lab quality management indicators also for us very important, which we need to receive from the customer clinic. Optimized oocyte towing protocol establishment. This is really important. Of course, each media, brand media has their own protocol, but each egg bank have their experience, big scientific experience, and also from another side, a practical experience. So we update and optimize the protocol according to our egg bank. Of course, it's for us very important to establish this protocol also our customer clinics. Video call and physical lab audit. Of course, now because of pandemic, physical lab audit is more difficult. So we are trying to much more with video call. So anytime when our customer clinic has a difficult situation or question or any process which we can help them, we can just have with them direct video call and to help to share all of our information. So online support during towing process, also this quite often we are doing. So when they have the towing process, when they start to tow the oocytes, if they see some difficulties in a first stroke, okay, next stroke we are trying to tow all together. We are trying to share all key factors of what they can help to them to increase their outcome. And of course, we have also still embryologist training in egg bank for the supplier clinic. Still all embryologists from our customers, if they have any question or if they have any wish to come to our lab and to get kind of training for warming process, of course, we are ready to accept this. So this is our KPI form, which we are asking from each customer clinic. There are different questions which we will evaluate their lab quality process and which is really directly affecting the quality of the process for egg towing, cultivation, and blastulation rate. So what advanced technologies we have in egg banking? Zona pellucida detection by pulsoscopy, Meiotic spindle detection by liquid crystal technology, artificial intelligence-based oocyte morphology assessment, time-lapse imaging and video during oocyte vitrification process, donor oocyte polar body NGS testing. So zona pellucida in donor oocyte, why for us is important? So we are using for that pulsoscopy system, which we can evaluate zona birefringes. So this is really important because when we have the high zona birefringes, always we find out high pregnancy rate, clinical pregnancy rate and implantation rate than low zona birefringes. So we can evaluate this one only by pulsoscopy techniques. Meiotic spindle determination. This is a really important for us because according to our research, we find out when we find exactly visibility of the meiotic spindle, we get a better outcome because abnormal meiotic spindle affect chromosome configuration and increase aneuploidy rate of oocyte as a complex aneuploidy. Also, each donor oocyte are observing before vitrification process and location of the spindle captured by time-lapse camera with the local navigate place, especially when I will talk next slide about polar body NGS testing. Before ICSI process, sperm injection process, spindle location is really important. So this is a typical image what we are giving to our customers when we detect meiotic spindle localization. So according to this time-lapse picture image, they just directly use this image as after towing and they do fertilization process. How we are using artificial intelligence in donor oocyte selection. So different parameters, different information coming for the artificial intelligence by the fiber optic crystal time-lapse camera. So polar body size, oocyte diameter, perivitellin space, cytoplasmic activity, and thickness of zona pellucida. So all parameters collecting by artificial intelligence. And then we have our direct parameter, which artificial intelligence can get as a reference parameters and can compare and give us which oocyte has a really quality one, which can go directly for egg bank. Oocyte is going to egg banking, and other oocyte we are using for fresh donor cycle. This is how artificial intelligence making a selection and show us that there is the different kind of damage on the donor eggs as solar or polar body damage or cytoplasmic granulopathy. Even if we have many different embryologists, so artificial intelligence still can get a data from the different embryologists and can collect on the main computer and can give us exactly result for each embryologist. So this is the algorithm how we are using on the calculation, mathematical calculation on artificial intelligence, how we are calculating diameter of cytoplasma, diameter of zona pellucida, also thickness of zona pellucida and perivitellin space. This is the difference between artificial intelligence and embryologists. So almost as number of grade 1 oocyte, there are 10% difference between artificial intelligence and embryologists. Why we are doing also polar body biopsy testing? Because still we believe 20 or 30% of donor eggs still has aneuploid. So that's why we are trying to get maximum euploid oocyte and to give our customers more chance even if they have low number of eggs. So mostly the customers are buying like 6 or 8 eggs, but still from the 6 of 8 eggs, still possibility 2 or 3 eggs can be aneuploid. So we cannot analyze this if we are not doing a polar body testing. But while we are doing polar body testing, we can give the customer directly 6 euploid oocyte and can increase the chance of euploid blastocyst, of course. So this is our outcomes between euploid oocyte after NGS, polar body NGS testing, and euploid blastocyst after fertilization and cultivation. And if we find out from the euploid oocyte, almost 89% of oocyte from the first age group has euploid blastocyst. From donor 30 to 32 years old, 85, and young donor group, almost 91% of the blastocysts which we receive from euploid oocyte that they are already euploid blastocysts. So still this is a big chance for the patient. So polar body NGS testing, firstly, genetically tested oocyte is an advanced technology which may increase chance of euploid blastocyst outcome and pregnancy. Polar body biopsy does not affect oocyte morphology and survival rate with the optimized vitrification and thawing process. Genetically tested oocyte has significantly better euploid blastocyst rate and implantation outcome, and chromosomal abnormality risk may decrease after the polar body NGS testing. Finally, Ovogene Egg Bank can supply high quality and totally transparent supplier clinic and embryology services, active laboratory audit system and quick response, optimized vitrification and thawing protocols, which is unique for Ovogene Egg Bank, advanced technologies which will increase the efficiency of egg donation program, scientific background and embryologist training programs for the customer clinic, available embryology and genetic counseling for egg banking and for whole embryo process, not only for thawing or like performing ICSI. We are following our customer clinics till the pregnancy. Thank you very much for your time. If you have any questions, so we will be ready to give answer.
Thank you so much, Birol. Dear colleagues, thank you for your comments. I have seen the comments. Lindsay, hi. It's good to see you here. If any other questions, please let us know, or any points for the discussion. I can see a lot of our partners here. Any feedback, any comments, anything you would like to share with us, please do. Also, as you know, we are available 24/7 for all your questions, any comments, any discussions, etc. Thank you also for our team members who joined our webinars to support it as well. So we will be happy to see you next month. We will announce the next topic of our webinar, and we'll be happy to keep sharing our results, achievements. Actually, something new what we can say, yesterday or today we performed the next cohort of the oocytes for NGS testing, which is going to fly over to Hong Kong soon. So we will control this process, and we will keep fingers crossed for the clinical and embryology team there to provide all the processes according to our recommendations and to maximize the patient result with the NGS tested euploid oocytes. Anything else, Birol, to share? Actually, we have a question. The first question.
I can see on chat we have one question: how to perform fertilization to eggs without polar body, why we are doing NGS testing. That's why I try to explain in different advanced technologies why we are using. So when we take the polar body for NGS testing, and when we freeze the oocyte, of course, still after thawing, we are making a single shot of laser. It will be difficult to identify where is the hatching area, but still we will give them active time-lapse picture, so which will show a direct localization of the polar body where it was before, and also direct localization of spindle. So we will give them a direct graphic, is a live image, is a direct from time-lapse. When they thaw the oocyte and they put under the microscope, they will identify so easy and quickly. Of course, for ICSI still, there are a different process than usual ICSI. We are giving them a special protocol during ICSI, how they should work. Also, we are giving a special type of ICSI pipettes, so what they need to use. That all protocol is going when they receive NGS testing oocyte, we are sending all material and protocol together with NGS tested oocyte. So they don't need to worry. We already tested with many oocytes. We didn't find any kind of damage or problem during the fertilization. And we have optimized warming protocol. Also, they will not have any problem during warming.
Thanks, Anastasia, for your question, and Birol for the answer. Anyone else would like to ask the questions, please do. We are still with you for a few more minutes. Even this is late for many of you. We know that, but this is a good time to talk after the hard working day. Great. So thank you all for staying with us, and we will be happy to see you again during our webinars at Ovogene Egg Bank. They are all recorded, and you can find the videos on our webpage and also on the social medias. So we are always in touch. We are always happy to communicate with you, to share, and we will keep working on our scientific and practical experience to get more cases to be shared with you, more topics to be discussed, and more learning from each other. Thank you very much, and enjoy your evening. Stay safe, and see you soon. Thank you.